Okay, so we are going to talk about now the JNK signaling pathway. So remember, in the two previous one, we saw that they have a similar architecture. So you have a receptor in both. So it's a signal transduction, and then there is a changes in the gene expression in the nucleus. And in both cases, it can do several things, but include cell proliferation, cell growth, and of course invasion. Now we're going to talk about the completely opposite, that is the JNK pathway that is involved in cell death. So when we talk about the JNK pathway, here the key is that this is a pathway related to apoptosis, and here you see a normal cell that if you see the cell membrane is surrounding, there is no invaginations of the membrane. Here you can see that invaginations of the cell membrane start appearing, all the organelles start getting closer to each other. Later you start forming these bubbles, these spheres, so the membrane start bubbling, and then if you see pieces of organelles will start getting into those sections and to those tiny particles that, remember, in the cell they don't waste anything, so some of them will be eaten by macrophages, other one will be, everything will be reutilized, and so all the nucleic acids, all these phosphate, they will be all these molecules will be used again. Okay, so these are four stages of the apoptosis from a morphological perspective of the cell. In the following sections, I will tell you a little bit more of the pathways involved. You don't need to memorize it. I just want that you see all those molecules that are involved that we have been characterized, and you might hear some of them in the future lectures. So key important thing, remember that this, let me delete this, that the JNK pathway is involved generally in cell death. And what happened generally is that, for example, here there is an external signal. It can be UV light, it could be gamma radiation, it could be so many things. And here you will find proteins. Yeah, you will find a receptor protein that it will affect uh, specific proteins that are called caspases, very important, and those caspases are key, are fundamental for the process of apoptosis. Okay, so remember, you have an extracellular stimulus that it could be, for example, UV radiation. There are so many examples. And why you can do that, if you have UVA radiation, you will have problems in the nucleus. You need to avoid those problems. So what happened is that the cell activate those caspases and say like this cell has to die. Why? Because if you have UV radiation in the cell, this is how mutations are. They were built in the past. You know, they were made how people used to make mutation in fruit flies. So. So what we do ourselves, what they do is let's try to avoid all these DNA mutations that they can produce cancer, and they activate these proteins that are called caspases, yeah, that are proteolytic enzymes. So what they are doing, cutting, yeah, they're cleaving specific proteins in the cytoplasm and the nucleus that trigger the cell death. Okay, so I hope that from this very general perspective, you remember JNK pathway, that signal, and these proteins caspases are very important. Please don't get scared. Yeah, these are more pathway, but it's the same thing. So you have a receptor outside, yeah, and I will show you later the receptors, that then you have a transduction, that later you have the nucleus. Good, so what is going to happen, that you have this JNK, here you see again phosphate is the same idea, and it's going to the nucleus, and here it could be an increase in the death factors. Yeah, so if you turn on the apoptosis, they will be, the caspase will be active and cell death, and it will trigger cell death. In contrast, the opposite can happen. You know, you can turn it off, and you can, this survival factors will be active. Yeah, and look, so now this is this is the opposite pathway, and this is how you can control one or the other one, that the cell survive or the cell die. So when you have a mutation in this pathway, so let's delete this. When you have a mutation here, what happens is if you have a mutation, for example, this protein, what you're going to do is that you will have off the caspases. So there will be no no cell death. And what else could be happening? That if you also have a mutation in these survival factors, now they will be always on. And if they are always on, so you're going to have no cell death and a lot of proliferation. And this is one of the cases where, that cancer definitely can appear because you have too little cell death and too much proliferation. Now I'm going to, in the following slide, I'm going to go even in more details about the proteins. Again, you don't have to memorize everything. I just want that you know more about this. I'm sure that some of you already presented some papers related to apoptosis, or will present papers in turn that talk about apoptosis. So remember, here you have a dead receptor, same idea. You have a transduction here that later will affect the nucleus, the transcription. Here you have again the JNK pathway. What I wanted to show you is that again phosphorylation is very important. There are many proteins, and here you have the activation of the caspase. There's not one caspase, there are different type of caspases depending on different type of cell death, and we will talk in the future about this BCLA that is important. BCLA2 is important for this death signal, and the mitochondria in some cases of apoptosis is also important. Okay, so again, just to give you a big picture that now how we cure cancer. So we need to activate and reactivate some part of these pathways. Yeah, it's like if you go from Winnipeg to Alberta, and there are to Edmonton. So here you can go take this highway, and if something is broken, you need to fix it, or you need to take another street to fix it. And this is what we do with cancer treatments. Try to repair some of these proteins, signaling pathways that are not working.